Hi, this is Alex from devstories.eu. Biopython Fundamentals, Episode 7 of 18. You need to download 10,000 sequence records. If you write a standard web scraper to hit the NCBI website, your IP address will be blocked before you finish the first 100. The National Center for Biotechnology Information explicitly forbids automated scraping. To pull data legally and reliably, you need to query NCBI databases programmatically using bio.entrez. Before making any network requests, you have to follow the NCBI API guidelines. First, you must tell them who you are. You do this by setting the entrez.email variable to your actual email address. If your script goes out of control and overloads their servers, they will use this email to contact you before permanently cutting off your access. Second, you must respect their rate limits. NCBI restricts unauthenticated users to 3 requests per second. Biopython handles this automatically by enforcing a delay in the background. However, if you attempt to launch multiple scripts in parallel to bypass this delay, NCBI will detect the abuse and ban your IP. Play by the rules. The workflow for extracting data from Entrez usually requires a 2-step process: finding the unique identifiers and then downloading the actual files. You start by searching with Entrez.esearch. Suppose you want to find the MATK gene in orchids. You call eSearch and pass it the database name, which is nucleotide, along with your search term, like orchid organism and MATK gene. This search function does not return biological sequence data. It returns an HTTP response that acts like an open text file containing an XML document. Here is the key insight. You do not need to write a custom XML parser or extract strings manually. You take that network file handle and pass it directly into entrez.read. This parses the XML and translates it into a standard Python dictionary. Inside that dictionary, you simply look up the key called ID list. This gives you a Python list containing the exact GenBank identifiers that match your ORCID query. Once you have that list of identifiers, you move to the second step, downloading the full records. For this, you use the entrez.efetch function. You pass it the same nucleotide database name along with an identifier from your search results. You also need to specify the format of the data you want back. To get a standard GenBank text file, you set the retrieval type argument to GB and the retrieval mode argument to text. Just like the search function, efetch returns a network stream, not a raw string. Because this stream mimics a local file, you do not need to save the downloaded data to your hard drive first. You can hand the network handle directly to seekio.read, specify that the format is GenBank, and it immediately parses the stream into a Biopython seek record object. You now have the sequence and all its biological annotations loaded into memory. When you are done, close the handle to free up network resources. The single most important habit when querying NCBI is treating your API responses as file streams rather than text strings. Passing those streams directly into entrez.read or seekio.read avoids unnecessary memory overhead and keeps your automation pipelines perfectly clean. If you find these episodes helpful, you can support the show by searching for DevStories EU on Patreon. Thanks for listening. Happy coding, everyone.